Alright, let's talk about protein metabolism. Protein metabolism is first going to start in the stomach. And in the stomach, we're going to have two cells that are going to play a big role in the digestion of proteins. One of them is called chief cells, and chief cells are going to produce pepsinogen. Pepsinogen is a zymogen, meaning that this enzyme is not active yet. In the stomach, we also have parietal cells. Parietal cells produce hydrochloric acid, which decreases the pH of the stomach. Now, when the pH of the stomach decreases enough, pepsinogen is going to be converted into pepsin, its active form of the enzyme. Pepsin is then going to start metabolizing proteins. This happens in the stomach, and this is where initially the digestion of proteins began. Now, as you may recall, bolus is turned into chyme in the stomach through chemical and mechanical digestion. Once chyme enters the duodenum, there's going to be two big hormones that are going to be released. In the duodenum, we're going to have the eye cells. These eye cells are going to produce cholecystokinin, which is a hormone that is going to play a big role in the metabolism of fats. We also have S cells. These S cells are going to produce the hormone screenin. Now, the hormone screenin has two different functions. One of the functions is to stimulate the liver to produce more bile, which is important for the emulsification of fats. And the other function, and the most important function, is to control the pH of the duodenum. This is very important because our pancreatic enzymes need a certain pH level in order to function appropriately, and secretin is going to be the hormone that is going to be doing this. Now, secretin is going to control the pH of our pancreatic juices. It is going to increase the pH of our pancreatic juices, and the way it is going to impact the pH is by two different mechanisms. So the first one is that it is going to stimulate chloride bicarbonate antiporter that is found on the acinar cells of our pancreas. Here is the cell membrane, and here's the intracellular space of the acinar cell, and this is going to be the lumen of the pancreatic duct. Here is a antiporter, and it is going to exchange chloride for bicarbonate. Right, and the purpose of this is to increase the pH of our pancreatic juices. In our pancreatic juices, we have enzymes. Alright, and these enzymes are going to be extremely important for the digestion of proteins, carbohydrates, as well as fats. This is why when we have a pathology in our pancreas, we're going to have malabsorption, because these pancreatic enzymes are crucial for the proper digestion of fats, carbohydrates, and proteins. Now, the second way secretin impacts the pH is through decreasing gastrin release in the duodenum and the stomach. Alright, so what produces gastrin? Well, we have G cells in our stomach and as well as duodenum, and sometimes even in our pancreas we have these G cells, and they produce gastrin. What stimulates the G cells to produce gastrin? There's going to be two different ways that G cells are going to be stimulated to produce gastrin. One of the ways is through parasympathetic innervation through the vagus nerve. So the vagus nerve is going to release gastrin releasing peptide, which is going to stimulate G cells to produce gastrin. Also, in our stomach, here's our stomach. Once proteins enter our stomach, it is going to in turn act on G cells to stimulate gastrin release. Now, what does gastrin do? Well, in the mucosa of our stomach, we have these cells. These are called enterochromaffin-like cells. These cells have a receptor, and this receptor is called the cholecystokinin B receptor. Once gastrin binds to the cholecystokinin receptor of the enterochromaffin-like cells, it is going to result in histamine release. Now, we talked about how in our stomach we have these parietal cells that release hydrochloric acid to decrease the pH of our stomach. Now, histamine is going to act on our parietal cells to secrete hydrochloric acid to lower the pH of our stomach. Gastrin can also directly stimulate the parietal cells for this. It does not need to just act on enterochromaffin-like cells to produce histamine, which is in turn going to stimulate parietal cells. It can also directly stimulate parietal cells by itself. Now, secretin is going to block this. Alright, so secretin is going to come in and block the release of gastrin in the stomach as well as the duodenum. One other important thing that we need to touch on is that in the parietal cells, gastrin is going to put these transporters on the membrane, and so these transporters are going to pump hydrogen out to the stomach, and potassium is going to come into the cell, and this is how it is going to acidify the stomach. Now that we talked about secretin, let's talk about cholecystokinin. So cholecystokinin, you can also think of it as having three functions. One of the functions of cholecystokinin is to act on the gallbladder. Right, it is going to contract the gallbladder for the release of bile. Cholecystokinin also acts on the acinar cells that is found in our pancreas to release pancreatic juices. Right, so we're going to contract the gallbladder to release bile, and we're also going to stimulate the acinar cells in our pancreas to release pancreatic juices. And the common bile duct over here is where the bile travels, and the pancreatic duct over here is where the pancreatic juices travel. And the meeting point of the common bile duct and the pancreatic duct is known as the ampulla of water, and this is located in the major papilla in our duodenum. So the third thing that the cholecystokinin hormone does is that it is going to relax the sphincter of Oddi, that is going to allow the bile as well as the pancreatic juices to enter the duodenum. And the bile is going to help us with the emulsification of fats, and the pancreatic juices, it is going to contain many enzymes that is going to help with the digestion of fats, carbohydrates, as well as proteins. So pancreatic juices have enzymes, and these enzymes are the proteases that help with digestion of proteins, that has amylase, which helps with digestion of carbohydrates. It also has lipase, pancreatic lipase, which helps with the digestion of fats. Now, the proteases that we want to first start talking about is trypsinogen, right? And trypsinogen is the inactive form of trypsin, right? And how does trypsin get activated? Well, in the apical side of our enterocyte, we have these enzymes called enteropeptidase. It is also known as enterokinase. And enteropeptidase is going to convert trypsinogen into trypsin. And one thing that you want to know about enteropeptidase is if you have an enteropeptidase deficiency, this could be fatal for the patient because they're not going to be able to digest proteins. Now that we have converted trypsinogen into trypsin, trypsin is going to convert inactive proteases into active proteases. Right, so we have several inactive proteases that are important to mention. One of them is called chymotrypsinogen. The other one is called procarboxypeptidase. And the other one is called proelastase. So trypsin is going to act to convert chymotrypsinogen into chymotrypsin. It is going to act to convert procarboxypeptidase into just carboxypeptidase. And then is going to act to convert proelastase into elastase. These are all the active forms of our pancreatic proteases, and they're going to all work with brush border proteases to convert dietary protein into oligopeptides as well as amino acids. Now, we have large peptides, small peptides, and amino acids from the conversion of the dietary protein with the help of active proteases and brush border proteases. We're going to have these smaller components, and these smaller components get to the enterocyte in different ways. Starting with the amino acids utilize the concentration gradient of sodium to bring itself into the enterocyte. So on the basolateral side of every cell in our body is a sodium-potassium pump, which is going to pump 3 sodiums out and is going to pump 2 sodiums in. So this is going to decrease the sodium concentration in the cell. So we're going to have a higher concentration of sodium outside the cell in the lumen. And we can use this concentration gradient to bring in amino acids. Now, on the apical side, we have a sodium hydrogen antiporter. So there's going to be hydrogen coming into the cell, and we're going to be pumping out sodium outside the cell. And we can use this to bring in hydrogen, to bring in small peptides into the enterocyte. So this should be sodium coming in and hydrogen going out. And so there's going to be less hydrogen in the enterocyte, so we can use that concentration gradient to bring in small peptides into the enterocyte. And large peptides are going to be able to enter the enterocyte through a process called transcytosis, which is essentially the engulfing of the peptide. So now that we have all of these smaller components of our protein, they can travel to the blood vessel, they can diffuse to the blood vessel, and then go to our liver for protein synthesis. Alright, so this is going to be the hepatic portal lane, which is going to take these to the liver where we can use it for protein synthesis.